Complement system is a group of plasma proteins that aid in nonspecific and specific immunity. These proteins are synthesized by the liver and are active when the pathogen enters the bloodstream. The proteins are usually represented by a C, then a number. There are three different routes in the complement system. The first is known as the classical pathway. Now, the classical pathway is antibody dependent, meaning that there must be antibody binding to an antigen of a foreign pathogen. The protein C1 or C1 complex, if you will bind to the antibody. C4 will come in and be broken down to C4A and C4B by C1 complex. The same process occurs for C2, forming the C4B2A complex, which is also known as the C3 convertase. The second route is known as the alternative path. This is an antibody-independent mechanism that occurs when C3 experiences hydrolysis, creating C3H2O. Factor B, which is another protein in the complement system, will cleave into factor BB and factor BA by factor D. Factor BB will bind to C3H2O, forming the C3H2OBB. C3H2OBB will cleavage C3 into C3A and C3B. The C3b will attach to factor BB, forming the C3b-BB, which is the C3 convertase of the alternative pathway. The third route is known as the lectin pathway. This pathway is activated when the mannose-binding lectin, also known as MBL, binds to a carbohydrate, usually mannose, which is found in the microbial cell surface, in which mannose-binding lectin serine protease 1 and 2, or just MASP1 and MASP2, will bind to mannose-binding lectin, forming the C1-like complex. For simplicity, we will call this the MBL-MASP complex. The MASP2 from the complex will cleave C4 into C4A and C4B, and the same process is seen for C2, creating the C4B2A complex, which, if you can recall, is our C3 convertase. Another molecule known as ficillin will have the same functions as the mannose-binding lectin, but ficillin can bind to different types of carbohydrates. In the complement activation, the C3 convertase created by the three pathways will cleave C3 into C3A and C3B. This produces four different end results. When C3 dissociates into C3A, C3A influences mast cells and basophils to secrete histamine and other inflammatory chemicals, which activates the action of neutrophils and macrophages to inflame the tissue. This in total creates inflammation. On the other hand, C3b binds to the antigen-antibody complex, in which red blood cells transport that complex to the liver and spleen, where macrophages will phagocytize the complex. This process is known as immune clearance. C3b can also coat the pathogen in a process known as opsonization. Essentially, C3b makes the pathogen tastier for neutrophils and macrophages, promoting phagocytosis. Lastly, C3b will bind to the C4b2A complex, creating the C4b2A3b complex, which is the C5 convertase. This convertase will split C5 into C5a and C5b, in which C5b will bind to C6, C7, C8, and C9 to create the C5b 6, 7, 8, 9 complex. This complex is known as a membrane attack complex, or just MAC for short. The membrane attack complex will attach to the pathogen to form a hole to expose the pathogen to the extracellular fluid, in which the pathogen will die since it cannot maintain homeostasis. This process is known as cytolysis, which is the most significant action from the complement system. Here's the end result of the cleavage of C3.